Okay, let's continue. What is our next paper showing here? It's something that was in Croatian Parliament on the 1st of December 2023, when we had guests here, and one of the guests was Kevin McKernan, who firstly discovered there was a contamination of these vaccines with plasmid DNA, and this paper is showing the same thing. Let's see next paper. Yes.
This is a very interesting paper, and at first glance, it might be easy for people to dismiss it because it was published in something called the Journal of High School Science. So, someone might say, well, high school students did this study. How seriously can we take it? But I'll get into that in a second. The title is A Rapid Detection Method of Replication-Competent Plasmid DNA from COVID-19 mRNA Vaccines for Quality Control. It was published in late December. Now, a couple of things here that kind of counter the argument that, oh, these are not, like seasoned scientists that performed this this study. First of all, the study was performed at a U.S. Food and Drug Administration laboratory, and it was performed under the supervision of FDA scientists. So these were not just three kids that were like in their, in their bedrooms running an experiment. They did this in an FDA laboratory alongside FDA scientists. They wrote a paper that was peer-reviewed, and the scientists that I spoke with said that the paper is methodologically sound. So, whoever these kids are, their advisors did a good job of guiding them through this process and helping them write this paper. So let's get into what the paper is actually talking about. The paper found that the levels of residual DNA, in other words, DNA contamination, in the Pfizer COVID-19 vaccine, so it focused specifically on Pfizer vaccine, were anywhere between 6 times to 470 times higher than regulatory safety limits. What are those regulatory safety limits? According to the FDA, it is 10 nanograms per dose. So the safety limits, according to the FDA, 10 nanograms of DNA material per dose is the maximum for a vaccine. And yet this study found that the contamination level was 6 times higher, all the way up to 470 times higher in this vaccine. Now, the study says that the potential health risk that these residual DNA fragments may pose to humans is still unknown. In other words, it's telling us further study is needed. But one of the things that the author said is that this DNA contamination may result in what they call insertional mutagenesis, in other words, DNA mutations. We talked about this before, that can potentially cause cancer. You mentioned Kevin McKernan before. He was, I think, the first scientist that identified this DNA contamination, and then you had some public health agencies, I think notably that of Canada, that confirmed that this contamination exists. McKernan said, I interviewed him a few months ago on CHD TV, which is the Children's Health Defense TV channel, that DNA in vaccines can pose health risks because the DNA can integrate into the human genome. It can disrupt the human genome, or it can disrupt other genes that can then, lead to the onset of the growth of cancer cells in the human body. Now, the final thing I'll say is that, again, high school students, even though they seem to have been supervised very well, they did the study, but there have been several studies in the last 2 years or almost 2 years that have found the DNA contamination in all sorts of COVID-19 vaccines that were sampled. So this is not an isolated result. It's another one of several results that studies have found showing this contamination.
Yes, and of course we have officials from outside of officials in Canada, not only in Canada, they found this. But just that they say it's not that danger, but there's something very important. This limitation of residual DNA was made much before they invented and used these nanoparticles, lipid nanoparticles as a vehicle to insert, exactly to come in this in a cell. And that means they have to reevaluate these limitations because we have now new technology. And it's not only that problem, there's an additional problem that was frame shifting. That means this messenger RNA send a message to produce some other proteins, and we don't know which one. And we have already 13 billion doses injected, and there was production of some other proteins, and nobody knows which one, because that was a mistake in this technology. They admitted, okay, we were building this airplane while we were driving this airplane, but let's start from beginning again and reevaluate again all these things, because this is completely new technology what we are using now in so many people, and there is so many mistakes, and we cannot pass by and just say, look, it was a mistake, we can repair it again.